Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class, we talked about looking at transcription factor binding sites or identifying histone modification sites using chromatin immunoprecipitation. So, we will extend that to much more general accessibility study and we will talk about chromatin accessibility in this class. Okay. So, this is the agenda for this class. So, we will talk about methods that can allow us to assess chromatin accessibility, and again the idea is very similar, right. We want to look at the role or the impact of epigenome on gene expression. Okay. So, chromatin accessibility is a much broader term. So, we are mostly looking at open chromatin and closed chromatin regions, right. So, as we have discussed in the introductory class, so open chromatin regions, so these are mostly transcriptionally active, whereas the closed chromatin regions, right, these are transcriptionally inactive, and this can actually help us understand or associate chromatin accessibility with the transcription process. Here are the keywords that we will come across. So, nucleus digestion, cross linking and fragmentation. Okay, so, we talked about chromatin immunoprecipitation experiments in the last class, and we also talked about the challenges that we face when you are doing this experiment. So, the first challenge is obtaining good quality and specific antibody. This is something that is very critical for the experiment. If you have non-specific interactions, it will bias your result. This is something we have talked about in the last class. One thing you should remember, right, or you should understand that if you want to identify, let us say 10 different transcription factors, you need 10 different antibodies against those, right. So, you have to generate antibody against each protein or each histone modification that you want to study, right, and this means this is a very tedious process, right. So, generation of antibody, that is also specific, and also this is quite expensive. Okay. So, what we can do about it, right. So, can there be alternate methods? So, we will talk about some methods. These are actually not actual alternates, but they can actually supplement these cheap experiments. Okay. So, and these methods actually look at much broader aspect, right, and they look at chromatin accessibility. Okay. So, these are 4 methods that we will discuss very briefly. The first one is called MNS-seq, the second one is called DNS-seq, the third one is FAIR-seq, and finally, we will talk about ATAC-seq. This is the most recent method for looking at chromatin accessibility. Okay, so, let us first look at what we mean by chromatin accessibility. As I mentioned, so we have open chromatin, and this is accessible to transcription factors, RNA polymerases. So, this region is transcriptionally active. On the other hand, you have closed chromatin, and this region is closed, means they are bound by nucleosomes, mostly occupied by nucleosomes and histones. So, they are densely packed and they are not accessible to transcription factors. Okay. So, open chromatin is transcriptionally active, whereas closed chromatin is mostly inactive. So, let us now look into the methods, the steps, and what are the different considerations for each of these methods. Okay. So, the first method that we will talk about is called MNS-seq or MAINE-seq or MIND-seq. So, this is the full form, micrococcal nucleus combined with sequencing, right. So, MNS stands for micrococcal nucleus, or we also call MIND-seq, micrococcal nucleus assisted isolation of nucleosome. Okay. So, this is again combined with sequencing. So, what this method does, it actually looks at location of nucleosomes and DNA binding proteins in the genome. Okay. So, one major difference from CHIP is that it will tell us, okay, which are the regions where DNA is bound by proteins, but it will not be able to identify those proteins. Okay. So, this is a major difference from CHIP. So, here we are looking at much more general or broader picture, but at the genome wide scale. Okay. So, just a very brief background about this MNS enzyme. So, this is first micrococcal nucleus was derived from Staphylococcus aureus, and because of its properties it was used to determine chromatin structure way back in 1975, and the term MNS-seq was coined in 2009. Okay. So, MNS-seq was first employed for determination of nucleosome occupancy in C. elegans in the worms. This was combined with NGS. Okay. So, here is the reference that you can look at. Okay, so when we talk about nucleosome occupancy, we are looking at where nucleosomes are bound in the genome. Again, this relates to open chromatin, closed chromatin conformation. And this method was then applied in human genome, also looking for nucleosome occupancy in 2008 in this paper that is mentioned here. So, how does MNS-seq work, right? So, this is where we understand the process, right. So, what happens in the first step is we digest the genome by micrococcal nucleus. Okay. So, this micrococcal nucleus is endo-exonucleus. So, as you see, this makes a difference in the process. Okay. So, MNS digests DNA in presence of calcium, okay, and it has slight preference for its consensus sequence, which is given here, right. So, it is an AT rich sequence which it prefers. And what we do is we actually at the end get the nucleosome bound regions, and we identify those by NGS. So, here is the schematic or the animation for the whole thing, right. So, we have this region here, okay. Again, some regions bound by transcription factors, some region are bound to histones, then these histones have some modifications, etc. Okay. So, what we do is we do extensive MNS digestion here. Okay. So, what will happen, because this is endo-exonucleus, it will cut, right, and it will also chew the ends, okay, and what you will you are left with are these bound regions, right. So, MNS cannot chew or digest these regions because they are protected by these proteins, okay, whether they are histones or some transcription factors, these regions will be intact. So, what we do then, we actually purify this bound DNA and then sequence, and at the end again what we will get is the sequences enriched for these occupied sites. Okay. So, some considerations for MNS-seq is that it requires many cells, right. So, we need about 1 to 10 million cells, that is a quite a high number. You need to titrate this process very carefully, right, how much enzyme you will add, how long you will incubate. So, all these things are very tedious, because with wrong enzyme concentrations or if you do really long MNS digestions, some nucleases it has been shown they are also sensitive to MNS digestion, right. So, even these nucleosome bound regions, they will be digested by MNS. Okay. So, there are some MNS sensitive nucleosomes. Okay. So, you have to titrate this very carefully. So, what we have seen that MNS-seq will actually map the occupied positions, right. So, it will not directly identify the active regions, but it will actually be an indirect determination of active regions, right. So, you know the occupied position, so rest of the regions might be free. And what has been seen is that it requires about 150 to 200 million reads. So, that is quite a large number for accessibility studies in the human genome. Now, moving on to DNS-seq, this is the second method that we are going to talk about. Okay. So, this actually helps us in direct identification of active region or open chromatin. So, this is in contrast with the earlier method MNS-seq, right. So, here we can directly identify the open chromatin region. Okay. So, the steps are very similar, the principles are slightly different. So, what we do is we digest by DNase enzyme. So, this is a non-specific endonucleus. So, it will cut indiscriminately, and it will not be able to cut at occupied sites. So, sites that are bound by histones or by other transcription factors, right, those will not be cut, but only the open sites will be cut. Okay. So, what researchers have seen, right, these active genes that have open chromatin conformation, they are more sensitive to digestion with DNase1, right. So, this means these open regions will be preferentially cut by this enzyme. So, here are the steps. In the first step, we subject the DNA to mild DNase1 digestion, okay. And then what we want to do is then we want to size separate these fragments, right. So, we want to select certain size that will be suitable for sequencing. Okay. So, to do that we actually take these DNA fragments and embed in low melt agarose plugs, and then these digested DNAs are size selected, right, and then we follow the library preparation and next generation sequencing. Okay. Again coming back to the animation, right. So, we have this DNA again bound by nucleosomes or transcription factors, but then you have some regions that are free, right, and these are the sites where DNase 1 will work. Okay. So, you can see these sites that will be attacked or digested with DNase 1, and you will end up with these fragments that are site selected fragments, right. So, we will select for some size that is appropriate, right, not too short, not too long, right. So, we will select at the end these regions that are actually open, right. So, these are not occupied by nucleosomes or by transcription factors, okay. And then we will sequence these site selected fragments, and they will be sequenced by NGS, and this will help us identify the open regions or open chromatins. Again, coming to considerations for DNase-seq, it again requires a lot of cells like MNase-seq, and you can then compare also with MNase-seq, right. So, this is also a time consuming process, and you need these enzyme digestions, etc., right. Again, you have to do the enzyme titration, how long you want to digest, what would be the concentration of enzymes, etc., and you also need about 20 to 50 million reads for standard accessibility studies of the human genome. So, this number is slightly less than MNase-seq. Coming to the third method, which is called the Fere-seq method, okay, and this method relies on cross linking. Okay. So, here you see formaldehyde assisted isolation of regulatory elements, and this is combined with sequencing. Okay. And this method also maps open chromatin region, and here is the original paper, right. This is describing this method. So, what are the steps in Fere-seq method? So, we have cross linking again of chromatin with formaldehyde, and this will actually capture the protein DNA interactions that are happening, right. And the next step is sharing of chromatin with sonication, okay, and then followed by phenol chloroform transformation. So, you have separation of aqueous and other layers, right. So, the DNA, free DNA will be in the aqueous layer, so which you can take out and sequence, and then the sequencing of the open chromatin regions, right. So, why I am saying we will sequence open chromatin regions, because when you do the phenol chloroform separation, if you take the aqueous layer, you will get the free DNA, which that is not bound by proteins, and once you isolate those DNA fragments and sequence, you will get the open chromatin regions. Okay. So, coming to the similar diagram or animation, again we have the DNA with nucleosome binding and transcription factor. So, we will illustrate, right. The first step is cross linking. Okay. So, again this is preserving this DNA protein interaction, because we are not doing any digestion here, right. So, we have this cross linking here. Okay. So, this will preserve this DNA protein interactions, and then we extract. Okay. So, we use the phenol chloroform extraction, and then we can separate out these two different types of genomic segments, right. The free region that are not bound by proteins, and the region or the segments that are bound by some protein, whether transcription factor or histones, and we can separate out this fragment that is free, right, just DNA, and we can sequence. Okay. So, the sequencing result will give us only the region that are open or free, right. They are not bound by any transcription factor or not occupied by nucleosomes. Coming to the final method, which is the ATAC-seq. Okay. So, this is the most recent method for assessing accessibility. So, the full form is assay for transposes accessible chromatin using sequencing. Okay. And here are two papers that actually describe this method, and as I mentioned, this is the most recent method, and one of the key steps in this ATAC-seq method is to fragment and tag the genome simultaneously by using an enzyme called TN5 transposase. Okay. So, what you have is that, so this is part of these transposons, right. So, they actually insert DNA in the genome. Okay. So, you can utilize that property by inserting adapters, right. These are the NGS adapters, right, that you can insert into the genome. Okay. So, these transposers, they actually insert these adapters and also fragment the DNA. Okay. So, that is why this is called tagmentation, right. So, you have fragmentation plus tagging, right. So, that actually is tagmentation. Okay. So, what researchers have seen is that because of steric hindrance, right. So, this is an enzyme, big enzyme, right, and it can only access the sites that are open, right. So, if you have like occupied sites that are occupied by nucleosomes or transcription factors and the open sites, this protein can only access the sites that are open. Okay. So, because of this steric hindrance, so all these adapters now will be integrated into the regions that are accessible, okay, because of the property of these transposes, right. So, it will integrate these adapters into the genomic regions, but where it can get access, right. And then what we can do is then we can amplify these regions, right, that contain adapters. So, we can use this adapter sequence as primer binding site. We can amplify these fragments and sequence using NGS. Okay. And this actually then identifies the open chromatin region, as we have just seen, because these integration of adapters will happen only in the open or accessible regions. Okay. So, here is the schematic again, right. We have the DNA bound to nucleosomes and proteins. So, imagine this is the transposes, right, and the orange is the adapted sequence, right, that will be integrated into the genome. Okay. So, you can by arrows I am showing the sites that are accessible to this enzyme. Okay. These are open regions, but the rest of them are not, right, they are occupied by nucleosomes or by other proteins. Okay. So, what happens here is then we get these fragments, right, that are also integrated with adapters. Okay. So, you get these fragments here, right, here you have this transcription factor that is there, right, and then at the end at two sites, right, you also get integration of this adapter sequence. Then you also have some fragments coming from these free regions, as you can see, right, and maybe this region here, right, these are free regions that are here, they are coming into the picture. But here also, like some of these regions that are containing only, let us say, one histone, they will also be isolated, right, they will also come in these fragments. And then of course, you will get a bigger fragment, right, because in this region there is no accessibility for this enzyme, right, because of steric hindrance. So, you will get this big fragment with the adapters at the end. Now, what you can do, you can actually site select some of these fragments, right. So, of course, the amplification will not work for very long fragments, right. So, you can amplify only certain range of fragments of so certain size, right. So, you can do a site selection also and then amplify and then sequence. Okay. So, once you sequence, you will get those fragments where the size is within a reasonable range, and these will mostly cover these open regions, right. So, open regions will be quite small because they are accessible for this enzyme, and it will integrate this adapter across this. The other regions, right, these ones, again this will depend, right, how long these fragments are and whether the amplification actually works. So, we can now imagine, right, if you have regions, genomic regions that are really dense, right. So, these are the closed regions with a lot of nucleosomes, there this enzyme will not have access or very rare access, and these integrations will happen at a longer distances, right, which will not be amplified by PCR, or you can discard those fragments by site selection. Okay. So, I hope this principle is clear, right. So, in this process we will identify the open chromatin region, right, that are accessible to the transposes. Now, here are some considerations for ATAC-seq. So, what researchers have seen that ATAC-seq can work on 500 to 50,000 cells. So, that is a very low number compared to the other methods that we have discussed so far, and this has recently been extended to single cell ATAC-seq. Okay. So, it means, right, you can now take out DNA, genomic DNA from one cell, and you can do this ATAC-seq. Okay. So, because this process works with very low amount of DNA, you can even work with single cell. This is not possible with other methods, right. So, other methods, they rely on some sort of digestion or chemical treatment, right, and one of the steps that you have with chemical treatment or restriction digestion is that you have to purify the sample after those treatments. Okay. So, you need to start with more samples, right, so that you have enough after those purification steps. Here you do not have to do any of these steps, right. So, there is no chemical process, right, chemical conversion or restriction digestion. So, you can work with even a single cell and take its genomic DNA and do the ATAC-seq. And the library preparation step is very fast here, right. So, this is 2-step library preparation compared to the earlier ones, which are quite laborious. You have to also titrate, right, which enzyme concentration you should use, etc. Here this is not really required, because this integration of adapters will happen only in the free regions, open regions, right, that are not bound by any protein. And again, this is not very highly dependent, right, on the concentration of the transposes. So, this process is very fast, right, and this is also quite easy compared to the other methods, and it requires about 60 to 100 million reads for standard accessibility studies of the human genome, right. So, this number of reads is comparable to the other methods, but the advantage lies in the first 2-step process, and we can work with very low amount of samples. So, this method has now been extended to single cell studies. So, we want to compare these 4 methods, and this is what I am going to do here. So, what we have seen is that MNS-seq, DNS-seq and FAIR-seq, they require chemical or enzymatic treatment on DNA for sample preparation. This is something we have talked about, right. So, MNS-seq, it utilizes this MNase enzyme. This has endo-exonuclease activity, and we map the only the bound sites, right, so, or the occupied sites. DNase requires this DNase1 enzyme, right, and FAIR-seq, it requires the formaldehyde cross-linking. Okay. So, this actually require chemical or enzymatic treatment. So, this could be this require optimization, and they increase complexity of the whole process. In comparison, ATAC-seq requires very fast 2-step sample preparation protocol. It utilizes this TN5 transposes, which actually integrates these adapter sequences, right. These are from NGS adapters directly into the genome and prepare them for library preparation and sequencing. So, another difference, major difference, is that MNS-seq, DNS-seq and FAIR-seq, they require more sample, much more sample compared to ATAC-seq, as I mentioned, right, because chemical treatment and enzymatic treatment, you need to purify or deactivate the enzyme. So, these purification steps means you lose some samples. So, you have to start with more samples. And what I have told you, right, ATAC-seq can be done on even single cell. So, it requires much less amount of sample, and you can do this on even single cell DNA, right. So, you will see these studies now coming out called single cell ATAC-seq or HC ATAC-seq. Another difference we have seen in this time is with MNS-seq, right. So, MNS-seq maps occupied or closed chromatin regions mostly, right. You have seen because of the properties of MNase enzyme, this chews up the DNA that is not bound by any protein or for example, histones or transcription factors, right. So, at the end we are left with DNA that is bound. So, we actually sequence the occupied or closed region. Whereas, the other 3 methods, the DNS-seq, FAIR-seq and ATAC-seq, they actually map open chromatin regions. Of course, they are not exactly identical. So, if you look at the results of these methods, you will see there are slight differences. Even though they map the open chromatin regions, you will see some differences across these methods. And also, you can compare these results with MNS-seq, and there will be some differences because of the properties, because of the protocols that we follow. So, these are the references for this class. So, to summarize, we have talked about different methods for assessing chromatin accessibility, and they do not require any antibody. So, we started with the limitations of the ChIP-seq experiment or ChIP-exo experiment, right. So, they required antibody, and these antibodies, they would have to be raised against each protein or each type of modification that you want to study, right. So, that makes the whole process expensive. So, we wanted alternative methods that could allow us to study these binding sites or accessibility. However, these methods that we discussed, these are not exact alternates because they can give us an overall view of the chromosome, right, the open chromatin and closed chromatin, but they cannot tell us about the transcription factor bindings, right, which transcription factor is bound where or which modifications are present at which locations, right. For that, we still need to depend on the ChIP-seq experiments. So, we have discussed these methods MNS-seq, DNS-seq and FAIR-seq. They require chemical or enzymatic treatment before library preparation, ATAC-seq relies on something called tagmentation process. This is tagging plus fragmentation, and this enables this insertion of these adapter sequences directly in the DNA, and this happens specifically or preferentially in the open chromatin region, because the closed chromatin region is not accessible to the transposase enzyme. Then we have seen that ATAC-seq can work with smaller amount of sample, right, and it works even on single cells. So, we have these single cell ATAC-seq data sets. And what I like to tell at the end is that we can actually by looking at this chromatin accessibility, we can actually start to connect the epigenome with the transcriptome, right. So, we know which regions are open or they are available for binding by transcription factors, or where RNA polymers can come and bind. So, this can then be associated with the transcription state of the cell. So, what we have so far discussed three different types of methods, right. So, we have talked about the methylation studies where we are looking at the DNA methylation patterns, okay. And then we talked about the chromatin immunoprecipitation method, where we are looking at binding sites of transcription factors or sites of specific histone modifications. And finally, we talked about the nucleosome occupancy. So, where we apply these methods to actually look at chromatin accessibility, or in other words, we are looking at nucleosome occupancy. We can differentiate between open chromatin and closed chromatin regions, and combine all these three together, we can connect epigenome to transcriptome. So, we have talked about these three different methods, right, three different types of methods that actually can help us identify or study three different types of epigenetic modifications, and these are all important for the transcriptomic status of the cell. In the next class, we will be talking about another aspect of epigenome, which is the 3D genome configuration. This is not any modification on the genome, this is actually the configuration of the genome itself and how it can affect transcription or expression of the genes. Okay. So, in the next class, we will talk about some of the methods that actually will help us identify the genomic configuration in 3D space, and then we will look into the different structural elements that are present in the genome. Thank you very much.